Okay, so now after some generation pass, a dramatic change in the bird habitat occurs, and they're talking about the survivability, like some individuals survive but some is not. So they mention about the relative fitness here, and it's given for us already. For the striped bird, the relative fitness is 1, so it seems that 100% on the striped bird alive. Okay, but for the speckled bird, it's not, it's just point A. So 28% alive and 20 is not. And now they ask us to calculate the new allele frequency. Okay, so it means that after like this change, the allele frequency has changed. So we need to find the new one. So before we can calculate the new one, we need to go from the initial one. So how can we find the initial one here? That we use the initial one that we got before, right? That 0.62 that we got from the beginning here, right? Okay, we go from there. So with that, and then we can see that this is for the recessive S, so that's why for the dominant big S, that should be just 100% subtract with this one, it means just that 1 subtract with 0.62, then this is for the dominant S, okay? And just like we know that for the homozygous, for the dominant big S, then it should be like the S go together, that should be P times P, right? That's P squared. And that's why you can find this value here by plugging in the values here. Okay, and then you do the same for the other genotype here, and that's why you can fulfill the first line here. Okay, this is the initial genotype frequency. So after that, we use the relative thickness that given. Okay, just put them here in the columns here. So what does it mean? It means proportional contributions to the population. It means that you type this value here with the relative thickness, then you can get the proportional contributions here. So for example, you got this value 0.144 multiplied with 0.8 here to got 0.115, and that's the proportional contributions to the population from this genotype SS. Okay, and then you do the same and you got these two values too. And finally, we find the relative genotype. So relative means what? Means that you compare this proportion contribution to the whole. So you sum them up together like this. It's just like you sum 0.115 with these two values here, the same like here. You got the total, and then you can find the relative frequency. A final answer is those values here. Okay. So what next? Yes, after we know like this is the relative genotype frequency after the selection. So once again we can see, okay, so after that, this is the value, right? And actually this is also contributed by the frequency of each allele itself. So now in order to find the frequency for the allele S, we can see that for the allele S, it's like SS here, right? We found it twice in here in this genotype and just once here in this heterozygous, right? So imagine if with this frequency, it means that it will be 131 individual in a total 1000, right? If you got like 131 divided by 1000, you will get the same ratio, right? That's the frequency, right? So the same if you got like 1000 in total, then the frequency for you to see the homozygous little LSOS recessive one is at 439. I just want to show you this step, and you can see why we can get to this final equations here to have it make sense. So why? So basically back to this one, frequency little s, it's just like you see it twice here, so it should be twice of that quantity, right? Because we are calculating the number of allele, okay? So 2 of it, okay? And then for that 429, you just see one allele. So this is the number of it in this hetero combination, okay? And there's no little s here, so we don't use that values. So this is the number of allele s in the sample. And then you divide it by the total, which is 1000 pairs in total. It means that twice of it is the number of allele because each pair will be have 2 allele, okay? So after that, you can see this is just about a math, and then you have the numerator, denominator, and they just have like 2 up and down, so we can cancel each other. And with this one, we don't have a 2 in here, so that's why this is 2 left here. So that's why you got this one as the result and not the final equation. Another way to look at this is that if you look at the genotype SS, you always find the allele, right? It means that for sure allele S will be in this genotype. So that means that the frequency, the certainty for the S to appear in SS is 100%. So it means that the frequency for this one is actually the frequency of this one. So be that value already. For the S to appear in the SS, like if you zoom in into the gene and then you just like check out one of allele, so there's a chance for it to be the little s and there's a chance for it to be the big S, right? So then it just like have 50%, okay? 50% to find the big one and 50% to find the recessive one. And that's why it's just like half of this one. So that's why the frequency for this one had to divide it by 2. And that's why we got this equation as well. So that's another way to look at it. And that's for the little s here. So conclusion, this is the new frequency for the little s, and then we do the same thing for the big S here. Then see, and then this is the value for the frequency of the speckle trait. Okay.